So there are five protein complexes present on the inner mitochondrial membrane. First is complex 1, that is called NADH dehydrogenase. They contain FMN, flavin mononucleotide, and iron sulfur. So they will accept the electrons and they will give it to the next electron acceptor. The next complex is complex 2, that is succinate dehydrogenase. Succinate dehydrogenase contains FAD and iron sulfur. So the FAD, when it is getting converted to FADH2, FADH2 will give the electrons to iron sulfur that is present in the complex 2. Now complex 3 is also called as cytochrome BC1. It contains cytochrome B, cytochrome C1 and iron sulfur. Complex 4 is called as cytochrome C oxidase and they contain cytochromes A, A3 and copper. Complex 5 is also called ATP synthase. Apart from these complexes, there are mobile electron carriers. One is ubiquinone and other is cytochrome C. So ubiquinone and cytochrome C are mobile carriers. They carry electrons from one complex to another. Now the electron transport system. First, in complex 1, NADH dehydrogenase. The NADH plus H plus gives the 2 electrons to complex 1, that is NADH dehydrogenase. And as the electrons move from FMN to iron sulfur and the electrons move towards ubiquinone, 4 protons will be pumped towards the intermembrane space because of the energy released due to oxidation reduction. The pumping of protons is possible due to energy released by reduction and oxidation of electron carriers FMN iron sulfur present in complex 1. Next is ubiquinone. The 2 electrons from the complex 1 goes to ubiquinone that gets reduced to ubiquinol UQH2 which gives electrons to complex 3, that is cytochrome BC1. The electron carriers cytochrome B, iron sulfur and cytochrome C1 gets reduced and oxidized in the process. The ubiquinol releases 2H plus to intermembrane space. So here the ubiquinol will be transporting or pumping 2 protons towards the intermembrane space. And when they do so, they will get oxidized and the electrons will be passing to complex 3. So from the UQH2 reduced ubiquinone, electrons passes to complex 3. The ubiquinol also receives 2 electrons from complex 2. Later, complex 2, the electrons from complex 2 also will be received by ubiquinone and 2 more protons will be pumped towards the intermembrane space. About complex 2, succinate dehydrogenase. When succinate, now this comes in the Krebs cycle, when succinate is converted to fumaric acid in the matrix, FADH2 is formed from FAD. The FADH2 gives electrons to complex 2. So when the FADH2 gives electrons to iron sulfur, the electrons will pass on to ubiquinone. But this time protons are not released because protons to release protons energy is required which is not sufficient. So electrons will go to ubiquinone and ubiquinone will get reduced and those 2 electrons will go to complex 3. The electrons will pass to ubiquinone and later into complex 3. Now complex 3 is also called cytochrome BC1. When electrons from ubiquinone passes through cytochromes B, iron sulfur and cytochrome C1 of complex 3, 4 protons are pumped into the intermembrane space and then the electrons will pass to cytochrome C. Actually in these 4 protons, 2 protons are as a result of NADH plus H plus, those electrons come into ubiquinone and the other 2 protons are because complex 2 has given to complex 3 through the mobile carrier ubiquinone. That's how 4 protons have come now to the intermembrane space. Apart from these, protons are also released by the ubiquinone towards the intermembrane space. Cytochrome C mobile carrier is present on the outer side of the cytochrome C is present on the outer side of inner mitochondrial membrane and carries electrons from complex 3 to complex 4. Complex 4 is also called cytochrome C oxidase, contains cytochromes A, A3 and 2 copper centers. The electrons from here will combine with oxygen, 2 protons, and they will form water. So oxygen is the final electron acceptor. As the electrons pass from one carrier to another, oxidation reduction takes place. Totally 16 protons will be pumped into intermembrane space, 10 from NADH plus H plus and 6 protons because of FADH2. The protons are pumped into intermembrane space that is more than the protons present in the matrix area. So this creates a proton gradient. Now comes the role of complex 5, that is ATP synthase. Protons from the intermembrane space diffuse into the matrix through F0 F1 subunit of complex 5 by the process of chemiosmosis. So the energy which is released in the process of diffusion of protons from intermembrane space to the matrix, that energy will be utilized to create ATP from ADP and inorganic phosphate. So NADH plus H plus and FADH2 are high energy electron carriers. They release electrons to protein complexes present on electron transport chain on the inner mitochondrial membrane. There are 5 protein complexes present and 2 mobile electron carriers. As the electron move from one complex to another, the energy released is used to pump protons across membrane towards intermembrane space. The electrons are finally accepted by oxygen to form water. Finally, as a proton concentration becomes high in intermembrane space than the matrix, chemiosmosis takes place. Protons from intermembrane space passes through the ATP synthase enzyme towards matrix, leading to the formation of ATP. And this is also called oxidative phosphorylation, as the complexes here they are getting oxidized and that energy is utilized to form ATP.